And whatever you see here is the live video from the lab. So not this.
He's going to have a split screen.
Dr.
Okay.
So Swami heads one of the divisions of clinical research, who he's looking after the whole process of translational immunology and molecular signaling.
Okay.
And start.
Thank you. So
we should finish on time.
Yes. So most of the work from transferring from the patients to the lab, most of them are based on tear biomarkers. Okay. So how does it look when we collect the tears after you collect the Shermas, when it goes back to the lab, and when we plot graphs like these? Where do they come from is what we wanted to show. And there are two ways it can happen. One is through flow cytometry. So we're going to patch in live from flow cytometry. It is a device which can measure close to 50 different cytokines from a single drop of tear. And Archana will be there on the other side. And of course, what we have also done is to make sure it is easy for the clinicians to have a kit. So we'll have live from both these units, okay? And we'll have one thing on the side to give you an idea of what that is. Flow cytometry is a cell's, facial recognition technology, you can say. So what you see there is a flow cytometer, and she is currently using a tear sample which is being analyzed using a process called bead-based multiplex ELISA. So what happens, this is a 2-day process, at least one whole day goes in this and another day of analysis. And this is the device, and what you see to the right side is the acquisition. Every cytokine gets measured like that before it forms like a dot or a bar in your graph. So, and we can also show that this is a laboratory intensive stage. We can patch in with the biomarker kit, where you can get the very same technology into your practice. And we can patch in with the biomarker kit now. How does it look? So right now you can see a Shermas which is getting shaked there with a buffer which goes into the kit. And what you see right side is a kit already loaded with a previous sample.
And then this kit can load anything. It can load, it's from your tears to CSF, blood, aqueous, vitreous, tumors.
Yeah.
So virtually, it's a pool, it's a well which can house anything in it.
Yeah. And no technical assistance required. You just add, and that's it. You get reliable, quantifiable data. This is both for scientific use as well as clinical use.
So this is something which is, there are. I discussed with a few of my friends from Heidelberg and AIOS that this is what helps us to look at the tears, and we were talking about how we can correlate this with your healing, basically epithelium or creating scarring or any of these factors. Of course, these cytokines here are limited, but I'm sure he's going to show us the images of the mass spectrometer. People asked, how do you, there are two things. One is you look at the cytokines, and cytokines are, you can have any number of cytokines, but you will have to figure out these cytokines are inflammatory driven, or they help in wound healing. But there are proteins which are the building blocks of our system, and that's where you look at a mass spectrometer.
We can patch the mass spectrometer.
Can you patch the mass spectrometer? It's very important because mass spectrometers, this is a mass spectrometer, very, very, it's the whole condition of how to keep this running. It is a very challenging machine. And it is at one given point of time, you can see thousands of proteins can be analyzed, and we are just going to upgrade our mass spectrometer to a higher one. So we call it as omics, O-M-I-C-S. Omics is metabolomics, lipidomics, then you have the proteomics and many other things. So it's like a mix of everything. So what happens is every gene, whatever the DNA, the mRNAs and all that, has to do a work. Genes will not go and start working out for you. The genes has to secrete certain amount of proteins. I'm sitting with very accomplished researchers here, so I'm still, pardon me, I'm trying to make it very simple because there are a lot of youngsters here. So it's important that these proteins are the ones which are messengers of the DNAs, and they produce, whether they produce tumors or they produce something which is helps to recover, is driven by the genes. And what happens is identifying those proteins, we call it heat maps here, am I right?
It is.
And
It is.
these heat maps have different signatures, and these heat maps, I mean, this is not, I mean, if there are proteins which can go into thousands and thousands of proteins, and when you have a heat map, there is a reference of how each protein is altered, and based on these proteins you can identify what type of a healing process is happening.
Indeed, indeed.
And this is something which is important for doing a lot of research work, because for example, I had a discussion today morning that there is a my friend here from IWIS, he said that we have bring in a different type of a laser which causes least amount of wound disruption. I said wound disruption is not driven by uh, by your clinical data, because you will not be able to make out anything, because whether you create more burns or less burns, your next day outcomes will be 20 by 20, because you can't see. Of course, confocal is one of the ways, but the real way of how gentle you are on a tissue is driven by how your proteins behave or misbehave. So when you have a misbehavior of your proteins, then your whole alteration of your process is different. So it is very important for technologies of companies which bring in these kind of lasers, whether diabetic lasers or non-diabetic refractive lasers, to see how gentle you are on the surface. Yeah. And the whole process is driven by proteins, and there is a group of people who are working with us who also deal extensively on the signaling pathways. That means each of these proteins have a signal.
Yeah.
And these signal, for example, there is a protein X which signals for it to do something. So in that heat map, you have to, from those thousands and thousands of heat maps, you have to find out what signal is driving and what kind of pathways. For example, in this is a driver. So what we try to do in our system is, we make the scientist to draw this kind of a pathway, and you can, if you can, I don't know whether you can zoom in those small genes out there. Excellent. If you can zoom in more, like for example, you can zoom that MEX12, ERK1, and alpha SMAs, multiple of these pathways. These proteins are the ones which you can see, and those are the ones which can cause fibrosis. And it is important that when you look at these kind of proteins like alpha SMAs and all that, these are the ones, for example, you do a fantastic lasers on your retina or any of those surfaces, you may have end up in scarring. Sometimes you do any procedure, nothing, scar doesn't happen, or your PVR, so multiple things. This is on the keratocyte, this is from a cornea, but the same thing applies in many other parts of the system. So the whole idea of what we are doing, the mass spec out there, is to pick up these
novel proteins.
So when you see this kind of a pathway out there, you are actually seeing those proteins being picked up. And then once the proteins are picked up, then we start looking back at what and how do we actually address these proteins. So this is a very interesting concept of wound healing. Again, we need this. So if you can go to the holotomography ones.
Yeah.
These are holotomography experiments. We have if you can patch the holotomography.
Yeah.
These are something which is excellent demonstration of, I mean, if you go back to your MBBS or even not even your biology classes, you would see a cell having, your mitochondria and all those images. You can actually see all of them. Earlier, to create a desiccation stress, that means to create a drier environment, you have to create you have to create in a rabbit model. And I was told when the first restasis was used, they had to stitch the lids of a rat, and so that the rats can't blink, the mouse can't blink. So when you can't blink, you create a dry environment. It was so barbaric in trying to find out whether it works or not. For example, here these are corneal epithelial cell lines. You create a desiccation stress. You can see that the cells are under stress, and then you use say a cyclosporin, rifetigrast, or one of the drugs, and you can see how it works. Interestingly, you can also see how say for example, how does the cyclosporin work on a scarring. TNF alpha is something which causes more of wound healing cytokine, and if you can enlarge that the patch there, so it will be interesting to see how we look. No, not that, the one from the live, the OCT of the cells.
This one.
Yeah, okay, if you can make it bigger.
Funny. The nano live on full screen, please.
Those are called nano live imaging, and it has actually bypassed us working on animal experiments for many of these things, because now we can real-time work on real cells. We also have some retinal pigment cells. We have a very beautiful tumor retinoblastoma cells. If I, Swami.
Yeah, I'm going to show you.
So, and we can virtually look at any cell, and you can create some. I was, you know, this is how the 3D cell explorer works. Swami can now explain.
Yeah, it is a holotomography. What you see here in a chamber is where you place your cells in a dish, and this chamber is controlled with humidity and the gaseous exchange which you would have for your normal cell to grow. And you have an objective with the fluorescence light coming through the objective end, and what you see rotating is the mirror which captures the whole thing in all directions.
Can show the screen also.
Yeah.
What you see on the screen, live screen. Show the screen, please. What is the output of the screen?
Yes.
No, not yours. The screen of
the lab. Yes.
And this is what he's. No.
I think it's getting swapped. Can we go back to the nano live monitor?
Yeah.
Yes. It is capturing, if you can see, the blue bars.
And
you can zoom out. Praveen, can you zoom out? You can get the entire frame. Yeah. So every few seconds, a picture at enormous depth is taken, so that you can have a 3D version of the entire cells.
We also have access to animal labs, and we have some imaging from the animal labs. If you can show how the imaging of the animal labs is. So that, you know, this is the work we do with and most of the animal related work is happening on the gene therapy part, and also on areas of
so we capture these kind of images from the animals. This is coming from a mouse, and this is the mouse cornea as well, but I'll show you how exactly it is done. We have an entire imaging suite, and we also have the imaging facility. We can't go inside, so we have taken a video, and you can see these various cages. We have cataloged animals with different treatments, with different age groups, and they have their feeds and bedding always checked. We have a veterinarian who takes care of their health, and we have to do with according to the IACUC protocol. So
the ethics committee of animals is more stronger than
the human ethics.
Thanks to Menaka Gandhi. I think it's very, very difficult in our country to do anything which is even, you know, unethical on an animal model. In fact, it's very strong.
Yeah.
And I think the team and Tirumalesh is very good with animal work. He's very gentle, and he does a lot of animal related work in this. And interestingly, we can also do an electroretinogram. I think Purna Chandra will start looking at ERGs on those animal models as we move on.
And this is somebody who is trying to get the anterior segment OCT live. The animal is alive, it's anesthetized, it's comfortable, it does not go through any kind of difficulty. And you can see the live imaging popping up there. And this is the live imaging of a posterior segment OCT, not from the same animal, from a different animal.
So we'll go to the gene therapy unit. So that's something which is, then the talk is also going to be followed by that. The gene therapy units have a very strict criteria in terms of who can enter, and because of the challenges of we work on gene modification. So there are different ways of how we are looking at gene therapies. It may look it is not too far away from reality. as I see by 2030, when we all be hopefully sitting in this place, I think lot of sessions will be dedicated into how therapies like these can change the treatment outcomes. So if you can go and patch the gene therapy units,
So we couldn't patch because
lot of these
we are not allowed to get inside because government has given the
Okay.
thing. So we have taken videos,
but that I think Pooja is anyway going to speak about the
Right.
corneal part of it, but
and
I
these
mean,
are,
can just discuss this a bit. I think this is what Tirumalesh showed, those
and we have a video.
you can explain.
So these are the various in vivo models which we have. Finally, the gene therapy product, the medicine, is going to go inside these animals. So we have to set that up. The AAV production, that is the vehicle which is going to carry the good gene, is produced in-house in the lab, so to make it affordable. So we have to make millions of it, vehicles. We call it vectors. Adeno-associated vectors are the vehicles, which is the most important part of the genetic gene therapy or the genetic medicine to begin with. So we can't just make it. We have to purify it to a level where we have to be absolutely sure that only the,
and its development requires a lot of
That's our
scientific
director.
involvement from a variety of different disciplines. That includes molecular biology, genetics, biochemistry, animal sciences, vector technology, and finally, the know-how to actually make clinical grade vectors. That's what we have achieved here in Netherlands, and the future is now.
So we definitely have come very close to it. Because of indigenous development, the cost is going to be a lot more affordable. Start to finish happens in one place.
Can you also ask somebody to patch up about storing sample? A lot of people
Yeah,
ask me
biorepository, yeah.
that how do we store samples? What is the whole protocol of storing a sample? Because that is what helped us to reach here. 20 years back, I give my credit to Dr. Kumar Menikavel sir. When he joined us as an advisor, the first thing he said was, don't throw
Biorepository.
anything. That time we didn't have any lab, and we didn't even have this. In fact, I thought this costs a lot. In fact, our slit lamp costs a good slit lamp costs less than this.
Yeah.
So I didn't realize this, but we were storing in Narayan Rudyalaya blood bank, and along with one slit, we had kept 5 years of thing. They threw it one day without our
Telling
notice.
us. Yeah.
Then we realized. So this hardly costs anything for people starting. This is like a like a Bank of Reserve Bank of India or Bank of London thing, and this houses thousands and thousands of samples. A lot of my ex-fellows and colleagues are here. Whatever they collected is all here and so well categorized. There are more than 30,000 odd diabetic samples.
Yeah.
We have close around
we crossed 80,000 to 1 lakh, sir.
yeah, and 1 lakh odd samples of everything. So and the question is, what do we do with this? See, out of those 80,000, there are thousands and thousands of pathways, thousands of new diseases, thousands of new pathways yet to be discovered. So biggest challenges is, for example, you can see that every corridor in our research, the 3 floors of research, every corridor has we only started with 1.
Yeah.
And now every 6 months we have to keep buying it. And how much does one of those storage house? How much does it house?
More than 1,000. More than 1,000.
And ask one of them to just open up one of the box. This is for a lot of
Biorepository, can you open one of the box which you just opened?
To get
please?
into this, my only advice is don't throw precious samples. You're doing a cataract on a RP patient, or you're doing anything which is interesting. Even if you don't want to buy, please find a place where they can house it for you. And if you can just go close up and just show how it is, it's like very well, each of those samples may contain a tear film, vitreous, aqueous, trabecular meshwork, sometimes even the CSF from.
Can you pull up one of the tubes, please?
This is gold. Kumar, even today, he says this is for a scientist. This is gold. How you keep it in your bank. And you go close up and see each one is labeled. Each one has a name, a number, and if you can, we don't have a QR system yet. But if you type this number,
Yeah.
you'll get the complete patient history diagnosis. I have at least found
You can close it.
at least 2 important genes in refractive surgery because
Yeah.
we had it, and what happened was we identified a novel mutation of lysyl oxidase in refractive surgery, and that became a big area of interest, and now that is the area of that because we had lenticule preserved in it. So housing your samples is very
Yeah.
very important, and I think that is the first thing which the researchers should do.
May I add something, sir? When I first joined Narayanath around 2015, 10 years ago, something very unique was happening. So they were all going through this biorepository to find out a batch of patients developed post-PRK haze, and you've been collecting the epithelium as part of the PRK and you've been storing it. Around the same time, they were able to see that this is before haze, okay, during PRK. A very unique signature was identified, and PREX1 turned out to be an indicator of PRK-induced haze predisposition. So that came in because of the biorepository samples. That was my first experience in GrowLab, actually, on how translation can be.
Can you patch in people working on corneal haze?
Yeah.
And
Yes.
and also people, Sangwan sir is here. I think we have not been able to achieve his level of
course.
thing in regenerative stem cells,
Yeah.
but at least somewhere close by with his inspiration. If you can just show the work happening on
Western blotting and qPCR.
yeah, and there is a team also working on biodiffusion. That means if
Oh,
you,
we have it, sir.
so biodiffusion is an area of interest where if you give that LFA1 antagonist, which Tirumale said, there is an eye drops on it. You know, I was telling them that if an eye drop, we need to know how much of that eye drop actually reaches the retina.
Biodiffusion.
And that is the whole work of biodiffusion. And if you give, say, people say, I give an omega-3 or a resveratrol, how much of it actually goes to one retinal cell? And there are various ways to look at these chromatography and those kind of work. And this is a work of biodiffusion. That this is something which is very important because sometimes we don't know how much whatever we eat, say an omega-3 fatty acids, how much of it actually comes to the corneal epithelial cells. And
That is Ravikran, he does the biodiffusion work. We can zoom out and show the
that's
HPLC.
okay. That's okay. In the interest of time, you can move to the haze work, corneal haze.
So
and there was a discussion with the Heidelberg friend from Heidelberg. We were telling that we can look at what are the haze forming, what are the surrogate markers in the epithelium, and how this can have an impact in the hidden immunology. And this is the qPCR work which we did.
We do both haze work as well as ectasia work. So we can patch in qPCR and Western blot.
Can make it a little bigger. No, not that. The guy
video,
on the other
yeah.
side.
qPCR and Western blot, please. Yeah, we can focus on the live. We can take the slides off.
Make it live, and the last one would be on iPSCs.
iPSCs, yes.
Pluripotent stem cells.
That's right.
And this is the work where you can see those graphs when you're studying those multiple genes. And
It goes off again. And Western blot also, if you can show. So what you see is a quantitative PCR machine, which is the quickest way. Now, because of COVID, everybody knows what PCRs are, but we're using it for epithelium stroma, any tissue which you want to have a quick breakthrough in the molecular pathway, which gives you the complication or a unique presentation.
So we move to the iPSC cells,
Yeah.
and we can end this session. That's okay. Is Kaushik around?
Kaushik is there, but I took all his videos.
So just quickly
Yeah.
show, just to complete it.
Kaushik, if you're there, please connect. Yeah. So any cell to any cell is what he does. He can you give him a corneoscleral rim or a blood, he can convert this to corneal epithelial cells, endothelial cells, retinal pigment epithelial cells, RGCs, neurons, whatnot. He's a magician there. And he's following a very important Nobel Prize winning method or concept called induced pluripotent stem cells. He takes the somatic cells, brings it to stem cell state, and pushes it to this type of cell you want to make. Functional indeed. He wants to do transplantation as well. So what you can see here is from blood, he has made keratocytes, he has made corneal organoids, he and his team, I should say, and he has made corneal endothelial cells, which is ready for transplanting for some of the dystrophies. And
interesting thing is about this is and organoids are creating virtual cells.
yeah,
You can create an organoid from melanoma or retinoblastoma. So instead of trying multiple drugs on creating a
In
model
animals.
on animals, you take that whole organoid, grow the cells from the same, say, retinoblastoma or melanoma, any cell lines, and start try different drugs. Gagan was working on it.
Yes, that's right.
And then see which one of these drugs works on this. So
And then-
organoid-based research to create models is a fantastic model of human diseases. I
Yes,
think
disease modeling as well as for pharmaceutical screening before we take it to the animals and further. And he has made a beating cardiomyocyte from a corneoscleral fibroblast. And that is a skeletal muscle which he has made, he and his team. All of these are for functional studies. If you can't get a primary cardiomyocyte, you can't get a primary skeletal muscle from a patient. So we use this technology. And he and his team has got neurons for us to interrogate some of the neurophysiology work.
beautifully you see how they connect to each cells. And that's how a thought process is done. Thank you, Swami.
Yeah.
Thank you. Thanks a lot.